Thank you. Thank you for inviting me to give this talk. So I'm a clinical scientist based in Birmingham, and I'm actually a microbiologist, so I'm not really medical at all. And the topic I'm going to talk about is biofilm-based wound management. So just an overview. So I'm going to set the scene, so might be a bit basic for most of you, but what are biofilms, why are they important, what's their clinical significance? And then I'm going to talk about biofilms in burns patients, because this is the group that I've been most involved with in my research. I'm then going to talk about some in vitro tests I've been doing on a couple of antibiofilming agents that are not actually antibiotics at all, they're completely kind of new agents. I'm then going to talk about comparisons with some dressings, and then I'm going to summarise and conclude. And I'm not going to talk about anything to do with biofilm diagnosis because we've already had a good presentation on that. And there's some really nice ESMID guidelines on this topic. So what are biofilms? So the definition of a biofilm is a structured consortium of microbial cells that are surrounded by a self-produced polymer matrix. And what this basically means is that instead of the bacteria surviving as just single-celled organisms on their own, what they do is they grow together in this community. So on the right-hand side, you can see this infected ankle with this horrible green slimy biofilm that has grown on top of the wound. And biofilms will grow on any sort of surface. It could be a natural tissue, it could be bone, it could be an artificial surface like a catheter. And they grow in these different stages. So from the left to the right, you've got these cells attaching to the surface. They then become mature and they recruit other cells through quorum sensing. You get this big community. And then on the very right-hand side, the biofilm is mature and it disperses new cells that go off planktonically to find new sites to make a biofilm. So where do we find biofilms? Well, we actually find them everywhere. So we find them in industry, on the bottoms of boats, in pipes. But in the human body, it's thought that 60% of infections actually do involve a biofilm. And these infections are mostly chronic because we don't manage to treat them effectively and eradicate them. And as I've said before, they're found in tissues. So you've got a range of different tissues. So endocarditis of a native valve, lung infection in cystic fibrosis patients, urinary tract infections, osteomyelitis. And then in terms of devices, you've got urinary catheters, artificial heart valves, and things like that. So what's their clinical significance? Why do we even need to bother about biofilms? So they're harder to treat. Because they live in a sessile state in this biofilm, the organisms are physiologically more resistant to antibiotics, and this can be up to 1,000 times. And in the laboratory, what we do is we tend to isolate the organism, so we pick one single species living planktonically, and we test the antimicrobial susceptibility. And this is a picture of an agar plate with the discs. So we don't actually test the bacteria as if they were living in a biofilm. We test them as if they're planktonic. So I think this is a key reason why biofilms are associated with treatment failures. They're also harder to diagnose because they're difficult to recover from clinical samples. This may be because the sample you take doesn't actually have the biofilm on it, or you may take the sample with the biofilm, but because the organisms are so used to living with others, they're more difficult to grow because they're not really used to being independent. The biofilms are also a significant burden of infection. So they're present in 60% of those people who have chronic wound infections, such as diabetic foot ulcers. They're associated with delayed wound healing. And if you have a biofilm, you have a greater chance of systemic infection and sepsis. So if we think about patients who have severe burns above 40% of their surface area, 75% of all these deaths are related to sepsis from burn wound infection. So going on to burns patients, which is really the main research, main focus of my research. So the mechanism of burn injury means that the burns patients are highly susceptible to colonization with bacteria. So they've got maybe large areas of open skin, they're immunosuppressed, they have lots of invasive procedures, and they have prolonged hospital stays. And in these patients, the organisms that cause their colonization or infection tend to come from their own endogenous sources, but also from the environment, water, air, and hands. And we've actually shown in our hospital in Birmingham that our water contains a large amount of Pseudomonas, which is a key pathogen of burns patients. So it's a big problem. So other key species would be Acinitabum, Bacter baumannii, and Staph aureus. So to sort of manage biofilms in burns patients, what we need to do is we need to minimise the colonisation of the wound in the first place in order to prevent a biofilm from forming. It's much better to prevent biofilms in the first place than to treat them once they've sort of occurred. So in terms of how we manage burned wounds, well, systemic antibiotics aren't actually very useful for burn wounds because research has shown that the antibiotic doesn't get to the area that it's needed at, so it doesn't go to the surface of the burn wound. So what we tend to do for burns patients is use topical agents such as washes or commercial antimicrobial dressings. And these typically contain silver, this is the main group, but also chlorhexidine, iodine, or honey. There's a range of different formulations depending on what your aim is. So it might not just be that you need to kill bacteria, you might want to absorb exudate or other things like that. And there's actually quite a lot of limitations of these antimicrobial dressings. So they have variable toxicity, they also have variable effectiveness, which I'm going to show you some in vitro data on. They're expensive. So in 2006 to 2007, the NHS spent £25 million on silver dressings alone. And this is a big proportion of, our budget. And a Cochrane review actually showed that there's little evidence to support the use of silver dressings, as generally they don't promote wound healing or prevent wound infection. So I've been doing some research to look at alternative topical agents that can be used on burns patients, predominantly to stop the colonisation of their wound with these bacteria. So I've been looking at a type of honey called Surgihoney. I've also been looking at acetic acid, which is the main component of vinegar. And there's others in the literature that I've not yet looked at. And these agents are good for antimicrobial stewardship because you're avoiding using systemic antibiotics in the first place. You're preventing, hopefully, a biofilm infection, so then you're avoiding having to use systemic antibiotics later. So in terms of biofilm-targeted testing, I've got 2 in vitro experiments that I perform in the lab. And the aims of these are to see if the agent can prevent the formation of the biofilm and to see if the agents can prevent expansion of a pre-existing biofilm. So going back to the diagram, we're wanting to stop those 2 main procedures. So this is the first experiment that tests if the agent can stop the formation of the biofilm. So what I do is I make an overnight culture of a bacteria. I then place it into a 96 well plate, and then to each well I place a similar amount of the diluted test agent, so honey or acetic acid. And this is in a range of dilutions from strong to very dilute. I then incubate this plate for 72 hours at 33 degrees C, which is the temperature of the surface of a wound, according to a paper that I read. And after 72 hours, I then assess for the biofilm formation using the crystal violet assay. And this gives you the minimum biofilm inhibition concentration of that agent that I've tested. The other experiment is called the MBIC, and this measures the minimum biofilm eradication concentration. So what you do with this is you form a biofilm on the underside of pegs of a PCR plate. So you take an overnight culture of isolate in broth, seed it into a plate, add a PCR plate on top, make a biofilm on the pegs of that PCR plate, and then you can transfer that biofilm into any test agent you want. And I've tested honey and acetic acid. Then after the correct length of exposure, you then place this peg plate into some sterile broth and you assess whether the biofilm is still viable, because if it's viable it will seed off planktonic cells that will make the broth turbid. So they're the 2 experiments. So on to Surgihoney. So this is an engineered formulation of honey which has enhanced antimicrobial properties due to the production of hydrogen peroxide. It doesn't rely on a single floral source, so it can be made easily and it's scalable in production. And it's been used clinically by Matthew Dryden and others, and it's had good outcomes with patients who have ulcers, pressure sores, caesarean section wounds, and other infected wounds. And I've measured the ability of Surgihoney to prevent biofilm formation on a panel of organisms as shown, and these are all key wound pathogens. And I've also tested a range of multi-drug resistant organisms as well. So this is the results of the experiment where I'm seeing if it can prevent biofilm formation. So along the x-axis, you have all the different dilutions of honey, going from 1 in 2 on the left-hand side all the way down to 1 in 4,096. And you then have a positive control, which is the amount of biofilm formed by the organism without any exposure to honey, and you have a negative control. And up the y-axis, you have the biomass of the biofilm that was produced. So for this experiment, you can see that the biofilm formation was reduced when you used Surgihoney dilutions from 1 in 2 to 1 in 8. And out of interest, I also tested Manuka honey on these same organisms. So these are Acinetobacter baumannii and a carbapenem-resistant Klebsiella pneumoniae. And when I tested Manuka honey, you can see that there was a slight reduction in biofilm formation at 1 in 2, but Surgihoney was a lot better in this experiment. I've also tested a range of dressings in this experiment. So instead of placing a volume of the test agent, I've placed a centimetre squared piece of each dressing into a well. And the results are the same. So you've got the depth of the biofilm that's formed up the y-axis, and along the bottom you've got all the dressings, and then you've got the Surgihoney. You've got the Surgihoney results here, and the positive and negative controls. So you can see that Surgihoney, when you use 1 in 2 or 1 in 4 dilution, and this is just in water, it reduces the biofilm formed, and it's comparable in its effectiveness to Acticoat, which is a silver dressing. An interesting Mesotran net here. This is a commercial honey-based dressing, and this was no better than nothing at all in this experiment in terms of biofilm formation. So on to acetic acid. So this is the main component of vinegar, and it has a long history of use through time. It's been used against the plague, ear infections, urinary tract infections. And it's actually used by quite a lot of centres worldwide in the burns field, but only on an ad hoc basis, so only really as a rescue therapy if other things don't work. So I've tested it against this panel of isolates, again containing multi-drug resistant organisms. And these are the results of the minimum biofilm inhibition concentration. So here you can see the same sort of results. Biofilm formation has been prevented from 5% acetic acid, which is the strongest concentration I test, all the way down to 0.16%. And you can see the positive control is very high, so there's been a marked reduction there in biofilm formation. And this is for a clinical isolate of Pseudomonas aeruginosa from the burns patient population. This is the result of the MBIC, so this is where you already have your biofilm and you're seeing if the agent prevents the seeding or reduces the seeding and expansion. And you can see here again, from 5% down to 0.31%, acetic acid did actually reduce the biofilm seeding. And then there's a period in the middle here where seeding was reduced but not prevented completely, and then there's a bit here where the acetic acid is too dilute to have any effect at all. And again, I've tested the dressings alongside the acetic acid. So here you've got all the silver dressings, you've got inodine, which is an iodine dressing, you've got the honey dressings here, you've got butchergrass, which is chlorhexidine, then you've got plain dressings, and then you've got the lowest effective concentration I found of acetic acid, which is 0.16%. So you can see a good reduction in biofilm biomass with acetic acid that compares to Acticoat and Mepilex AG. And this is for 2 isolates of Pseudomonas aeruginosa. So I just want to talk about the limitations. So I've shown that there's 2 in vitro tests that can be used to evaluate agents in the lab setting, but they're actually, in vitro tests are no substitute for in vivo testing, as natural biofilms are likely to differ to laboratory-formed biofilms in their physiology and how they behave. The experiments provide no information on the mechanism of action of these agents, and you also can't assess other important aspects, so toxicity and impact on wound healing. So to conclude, I don't think you can properly treat patients if you ignore biofilms, And I think in microbiology we're particularly bad because we perform antibiotic susceptibility testing on planktonic organisms. We know that biofilms are more tolerant of antibiotics, so I think it's a good idea to investigate alternative non-antibiotic agents that we can use first of all before we need to potentially use, conventional antibiotics. I don't think standard antimicrobial dressings are the solution at the moment, and I've shown that Surgihoney and acetic acid both have good anti-biofilming properties, and therefore they're good potential agents for new treatments and dressings, and this is nice translational work. But to take this further, we do need to do some controlled clinical trials. So I'd like to acknowledge the SRMRC, which is the research group I work for, the University of Birmingham and Aston University, because I get students from that university who help me do my work. And if you want to ask me any questions, they're my contact details. Thank you.
Thank you for a nice presentation. Are there any questions? Yes.
Well, thank you for this. Is a very nice study, but I want to ask what kind of strains of Pseudomonas you used, for instance. Are they the naive ones or multiple drug-resistant ones?
So there are a range of control strains and strains from patients, and they do differ in their antibiogram. There are a few that are very drug-resistant in there. Okay. And I've also tested all the 6 multi-drug-resistant organisms, so it seems to be universally effective against them all.
Thank you for the presentation. My question is, I think you have the comparison between acetic acid with the modern antimicrobial dressing. So a number of those antimicrobial dressings had a sustained release mechanism versus acetic acid. I wonder if you can comment on the the feasibility of using acetic acid for the management of biofilm in a wound environment, and using a compress or dressing or solution.
Okay, so I'm actually applying for funding to take the acetic acid work further. But the aim of the acetic acid dressing would be that we impregnate a gauze with the acetic acid that then gets placed on the wound. And actually, the experiments I've shown, I've done overnight experiments to see if acetic acid can kill bacteria, and they actually kill them outright. So as long as you've killed them, they can't form a biofilm because they're completely dead. And the aim would be looking at replacing the dressings twice daily, just while we're checking how effective the acetic acid is. But it would just be a cotton gauze that's impregnated to begin with.
Could I ask you where your Manuka honey came from, please?
The Manuka honey was the Convita, I think it was called.
So was that a medical grade one? Was it?
Yes, it's a medical grade one. Yeah, yeah.
Okay. Thank you.
Yes.
Klaus Kjeta from Denmark. I congratulate you with your study because it's in concordance with what I found. So of course I'm glad. And we also have a patent on it. So I'm really, really satisfied with your work. Just a few comments. You showed actually that there was a good result with some of the dressings with the silver. In our lab that I work with, they tried it with a broth with a lot of proteins, and the proteins will bind all the silver. And so, can you comment on that, because this could be the reason why it seems to work here, but in our test the silver compound does not work.
Yes, so I've not measured. All I've been measuring is biomass. I've not measured silver release, the impact of other agents, but that would be something I'd need to address.
Okay. And just to say that it works perfectly in patients too.
The acetic acid or
Yes.
the honey? Yes.
I use it extensively.
We do actually use it in our burn centre at a concentration of 2.5%, which I've shown is now too high. We could reduce it a lot more. Patients do report stinging, but not every time. But it does seem to be quite well tolerated.
So we use it in a 1% solution.
1%.
1%, and there is a gradient throughout the wound. So I think we need to have a bit higher percentage than is that's shown in the lab. Thank you.
Okay, thank you very much.